I love the word reappreciating. We're reappreciating STIs in the context of the HIV epidemics, and I think it's great that we're all here. I'd have to say I have no disclosures. I'm just an epidemiologist, so I don't get any money from companies. Any specific tests that I mention, I am not particularly endorsing. I also want to thank Rosanna, because she's given a great introduction to the things that we need to think about, not simply about point-of-care testing, but about diagnostics in general. So think back, remember Rosanna's talk, and we'll go forward and talk about new STI tests. Here's this map again. I want really to remind myself to say that the ones on the map are for curable STIs, and we have about 1 million new cases of those every day. And I'm going to talk about those. I'm going to talk about Mycoplasma genitalium, but I'm not going to talk about viral STIs today, so I'm sorry about that. What I do want to talk about is not just about diagnostics, not really very much about technologies, how about we prioritize tests and how we use new STI diagnostics. So in that context, two particularly important things are about the multiplexing of different organisms into the same test, and to really focus on antimicrobial resistance. Then I'm going to have a brief dip into what people think might be the future of STI diagnostics, and then come to some conclusions. So this is my opinion about what happens when tests, we try and prioritize what we need for new STI tests. And there's what we need and there's what we get because it's commercially viable. So what I think that we need is tests that address the places where there is a high burden of sexually transmitted infections. Those are low and middle income countries. What we get are, because it's commercially viable, are tests for where there is a high value market, and those rarely coincide. Going back to Rosanna's talk, what you would like are tests that are accessible, affordable, and accurate, and so for resource-poor settings, they need to be near patient with simple and robust technologies. What you often get is a laboratory-based test. It goes on an existing platform, and the platform has already been developed for something else, and then you're going to add some new targets to it. But the issue about STIs in developing countries is actually also that what we're looking for are tricky things to detect. And I'm going to come on to that when we talk about detecting antimicrobial resistance in new diagnostic tests. But what you get in the commercially viable and what comes onto the market are the low-hanging fruit, because those are the first ones to be developed. So we do have some promise in new incentives, because for the first time, in addition to having an essential medicines list, WHO has developed a list of essential diagnostics, and these are diagnostics that should be available in every country for everyone and available to everyone. And STIs were brought to the table to make this discussion, and I'm really pleased to say that in amongst the essential diagnostic tests for infections, we have several that are sexually transmitted infections. So there's a technological revolution that has occurred. If you're in the STI world, you will have known this for 20 years. It is the revolution of molecular diagnostics. And if you're thinking about developing a new diagnostic test, and it's going to be a molecular test, you have to have these 4 components in it. You have to have a specimen that you're going to test. That's maybe the easy bit, but not necessarily, because genital specimens and extragenital specimens aren't nice, clean things like blood. You've then got to have something that's going to extract your nucleic acids from the specimen, and then you've got to have a system that is going to amplify those nucleic acids, because you're trying to detect very, very small amounts of genetic material. And then you've got to have a fancy, nice, easily read system that's going to detect the product and then show you what the result is. So if you think about developing a new diagnostic test, and if you particularly think about wanting to bring that test near to patients with simple and robust technologies and fit it all into a nice small machine, that's actually quite a big challenge. So, Rosanna summarised for you point-of-care tests and focused on the syphilis and HIV tests. But of course, there's an overlap here between point-of-care tests and new diagnostics. So here I'm showing you what is available and what's in the pipeline for point-of-care tests to detect the bacterial sexually transmitted infections, chlamydia and gonorrhea, and trichomoniasis. The solid lines coming out from the green arrow are the ones which are on the market for at least one infection. And you can see that they're all neat, they're all lovely, they're all these beautiful little machines that we want to be able to put into clinics. The one I'm focusing on has the red square around it, is the Gene Expert machine. It's what Dr. Nicola mentioned just now. It is available for and being used for TB and for HIV, and this is a potential of a test that can and is being used to do research, but is not yet really being used for management because it's too expensive and it takes too long. It takes 90 minutes to do, and in many settings that's too long to wait for a result. But this is the sort of thing that we are aiming to achieve for STI diagnostics. So, the value and one of the technological capabilities of molecular testing is that you can detect, as you see with the chlamydia and the gonorrhea test in the Gene Expert, is you can detect multiple infections in one test. And that seems really, really promising. But we've got to be careful about this. So I want to just mention now Mycoplasma genitalium. It came up in conversation yesterday. Many people might not have even heard of this infection. And the reason we hadn't heard of it is because until recently, there were no commercially available tests to detect it. So there's this kind of circle that goes, if you want to find a new sexually transmitted pathogen, you've got to have something that detects it. So there were research tests developed in the early 90s, and now there are commercially available tests. We've got to be very careful how we use these tests, and I'll just leave that there for the moment. But I wanted to mention it now because the promise of multiplexing tests is that you could detect this test is suggesting that you can detect 10 STIs in one sample. Now, isn't that fantastic? You can come, you have a single specimen and detect 10 STIs. Fantastic. Well, let's just hang on a minute. So, firstly, if you're writing down lots of sexually transmitted infection names and you're developing a test, sometimes you forget to spell them correctly. I think that's a bad sign for advertising. Secondly, you have tests that are, here you've got organisms that cause urethral and cervical discharges, but you're promising that you're going to detect things that cause genital ulcers, and it's a bit hard to decide which specimen you're going to use for which you're going to want to detect all these 10 pathogens. The next thing is that just because you can detect an organism doesn't mean that you should. Some of these things aren't actually sexually transmitted infections, and we don't want to test and detect them, but we know that in many, many settings, detecting ureaplasmas and mycoplasmas is wasting enormous amounts of money and resulting in massive amounts of overtreatment. So we do want to detect some of these pathogens, but we don't necessarily want to detect them all in everyone. So let's move on to what I think is really the critical issue about diagnosis of sexually transmitted infections and wanting to have tests that are future-proof. If they're bacterial infections, they've got to detect antimicrobial resistance where that is relevant. Where is it relevant? Well, actually, fortunately, it's not relevant for chlamydia. And it's not relevant for syphilis, for benzathine penicillin. So we're counting those two out. The other three organisms, antimicrobial resistance is a problem. We know for gonorrhea it's a massive problem. But we don't have tests for resistance that are multiplexed into these jolly clever multiplex diagnostic tests. And we did pose the question, because molecular tests for gonorrhea have been available for nearly 20 years, was it not thought of then that incorporating targets for antimicrobial resistance would have been a good idea at the time? The one that we do have tests for is the new kid on the block, Mycoplasma genitalium. It is has a massive problem with antimicrobial resistance, but fortunately, along with the test for detection, there was also a test to detect resistance. So, Mycoplasma genitalium as a bit of a nightmare scenario. This is the infection that you haven't heard of. These are data, the black points are the data showing in Denmark and Sweden the proportion in research studies of samples that have resistance to azithromycin. Oops. So nearly 50% in Denmark and coming up for 20% in Sweden. And what we found is that for Mycoplasma genitalium, about 12% of people who start off with a susceptible infection, once you've given them their 1 gram of azithromycin, by the end of it, that Mycoplasma genitalium will have become resistant. So that's called de novo resistance. And our modelling study on the left shows that if we just carry on doing what we're doing, and this is in the absence of any commercial tests to detect M. genitalium, we're going to be reaching 100% resistance for an infection that most people have never even heard of. So a rational and a really good way of using new diagnostic tests is to say, let us detect the mutation that causes the resistance, and fortunately for M. genitalium, these have been reasonably easy to detect. And then we use that to guide therapy. So not only do we test people for the germ, we test for macrolide resistance too. And then we decide what treatment people are going to get depending on their result. If they've already got a resistant infection, you give them an antibiotic that's going to cure it. If it's a wild-type infection, you don't want to give them just azithromycin. But if you treat, these are mainly men with urethritis, if you treat them with doxycycline first, you bring down the bacterial load, and then it turns out that if you then give them a longer, bigger dose of azithromycin, you can reduce this emergence of de novo resistance by about a half. So that's what I would say is sensible multiplexing and taking into account antimicrobial resistance. So let's move on to gonorrhea, the major problem that we've seen for developing resistance ever since sulfonamides were discovered to be effective against Neisseria gonorrhea. The reason for showing you this very complicated slide is that it is actually very complicated to develop tests to detect resistance in Neisseria gonorrhea. Not only is gonorrhea just a super clever organism, it can develop resistance in multiple parts of its genome with multiple mechanisms of resistance. It has developed resistance to every antibiotic we have thrown at it. So this is showing you that it is difficult there are many targets that you would need to detect if you want to detect antimicrobial susceptibility for gonorrhea. And actually one of the problems at the moment is the tests that we do have, in many situations, probably about a quarter of them, we're not actually able to amplify and detect them. So these are real technological challenges for diagnostics. The one that is closest to being used in practice is a mutation in the JARA gene that confers resistance to ciprofloxacin. And you heard yesterday, and Rosanna's mentioned that if you could detect the ciprofloxacin susceptible infections, then you would be able to use that, and we wouldn't be wasting the ceftriaxone that we're hanging onto and hoping that gonorrhea is going to remain susceptible to it. I think that's a great idea, but that is a rich people's solution. And I just want to remind you that ciprofloxacin, for half of the world's population, a test to detect susceptibility to ciprofloxacin in gonorrhea is going to be of no use at all, because these countries have 70 to 100% of gonorrhea is already resistant. So we need to be detecting the harder to detect targets for the majority of the world's population. So let's have a peek into the future. I have to admit that we're not really in the present yet, because we haven't got the tests that we need to get to the populations that need it. But people are saying, and the people who develop the tests and develop the technologies, that what if you don't even just detect the target? Why don't we just detect the whole genome? Because we can. we can do whole genome sequencing. So then you could detect whether the organism's there, and you can detect whether there is any resistance target there for gonorrhea or for Mycoplasma genitalium. So there is this fantastic machine called a nanopore minion, not a minion, and it does whole genome sequencing in this really, really small little kit that you just put your sample in. Here you have to have a pure extract of DNA because you can't put in a pure a genital sample as it is. So you've extracted the DNA. It works away, it does all of the sequencing, and then it comes up with lots of complicated bioinformatics, comes up with your output of what is there. So we thought that's great. We want to evaluate this for gonorrhea. And we found that actually, whilst you can detect species with it quite accurately, given the tools that we have available, based on some clinical samples and the WHO reference panel of gonorrhea strains, these tools are currently available don't actually pick out the genetic resistance to targets accurately enough for us to be able to use it. But that is maybe going into the future. So I really want to finish by saying available new diagnostic tests, we are talking not just about the technology, but about how we use them. The ones that we have can't replace syndromic management at the moment in resource-poor settings, and in resource-rich settings, they could cause more problems than they solve by blindly testing many, many people who don't actually need that test, but using multiplex tests just because we can. So we need to use the targets that we want to detect. We need to choose them rationally. And we need to understand that what molecular diagnostic tests for antimicrobial resistance are absolutely essential, but they're really complicated. We actually need an incentive system that is going to influence development, like people are promoting for new antimicrobials. So I think these are financial incentives, supranational incentives for diagnostics companies, but the essential drugs list of WHO can also be used to say the tests that we really need. So I sum that up into saying if we want new diagnostics for STIs, we need the right test for the right infection in the right place. Thanks very much.